everyone. My name is Jeffrey Perera, and today I'm here with Ujjwal Rathore, a postdoctoral researcher in the Marson lab. Who are you, and what do you do?
Hello, Jeffrey. Hello, everyone. Thanks for having me. I'm a scientist in Alex Marson's lab, and I use gene therapy, gene editing to study viral infections. I mainly focus on studying HIV infection. So what I do is I use tools that can edit DNA or the genetic information, and then, infect these edited cells with HIV to figure out how HIV hijacks our system.
Tell me about your childhood and how you found yourself working at Gladstone.
So I've come a long way. I was born in Delhi in India, and I studied in a Hindi medium school. So my first language is not English. And in my school, I found some teachers who were really enthusiastic about answering our questions, and I used to have a lot of doubts. So I remember once asking my biology teacher a question that, we have same genetic material in all our cells, same genes. So humans have 20,000 genes. And I remember asking that if we have same 20,000 genes in our skin cells and same 20,000 genes in sperm cells and egg cells, why can't we make a new human from, skin cells? Mm-hmm. And my teacher had no answer to this question at that time, but he told me that maybe, we don't know answer to this question now, but maybe you will figure it out one day. And now I understand it's because different genes are expressed, although we have same 20,000 genes in each cell, but some genes are expressed in each skin cells and not expressed in, in them. That's what makes each cell different. Interestingly, Gladstone is a place where we figured out how to convert, fibroblasts or skin cells into any other cell type.
Who is one person that inspired you to pursue your interest in the sciences?
It's difficult to name one person actually, because what inspired me was really curiosity, just in general curiosity about things around me. For example, I was curious about why skin cells can't make new human beings. So it's just so there were two things which basically inspired me. One was curiosity, and the other thing was desire to bring change. These are things that inspired me to really come into science, and not any one person.
Mm-hmm. So how did you end up working with HIV and viruses specifically?
Yeah, so it's an interesting story. After I finished my schooling and bachelor's in Delhi, I ended up joining this institute called the Indian Institute of Science, Bangalore, which is the top research institute in India. And I was doing rotation in one lab in biophysics department, and I really liked what they were doing in the biophysics department. So what scientists in biophysics department were doing, they were trying to understand how proteins, which are the action molecules of the cell, how they take the three-dimensional shape, because the three-dimensional shape of a protein basically decides what function they're going to do, how they're going to function. So if a protein molecule has a shape like this, it might do a different function. But suppose it can't fold into this three-dimensional shape, it folds into some other shape, it might not be able to do its function that it does. So when I joined this department, biophysics department, and I was trying to understand how proteins fold, there were scientists who were trying to use this knowledge about protein folding to making viral vaccines. And I joined this lab, Professor Raghavan Varatharajan's group, where they were making vaccines against HIV and flu. And when I decided to join that lab, they asked me which project do we want to work on, flu or HIV, and I found HIV to be more challenging because it's been 40 years since HIV is discovered and we yet do not have a cure or vaccine for HIV. So I thought it's a difficult problem. This is what I'm going to work on, and more interesting. And I thought that probably this is where I want to invest my energy on.
So why is HIV research even important?
This is a very good question, because people often ask that although we don't have a cure for HIV, but we do have a treatment for HIV. So why are you people still studying HIV? And it's very important to understand this, because right now there are close to 40 million people in the world who are living with HIV, and not all of them have access to antiretroviral therapy. Even those people who have access to antiretroviral therapy, they need to take those drugs every day. So you need to take a pill every day for the rest of your life if you get infected with HIV, which is not always doable because of multiple different reasons. For example, just imagine imagine a scenario in which COVID pandemic is going on. You don't have to actually imagine. It's right. So
Right.
people sometimes say that right now COVID pandemic is going on. I would say that right now there are two pandemics that are going on, at least two. One is HIV pandemic and one is COVID pandemic. And because of COVID, there was disruption worldwide in supply chain, right? So we are all struggling with getting our reagents to do research in the lab. But now think about these disruptions affecting delivery of antiretroviral drugs. So it has been estimated by UNAIDS that many more people are going to get infected with HIV. A lot of HIV positive children are going to born because of unavailability of the antiretroviral drugs. And so I think it's an unsolved problem, which is affecting a large number of people. The drugs which people take to keep this virus, to stop this virus from replicating, they have their side effects. And as you grow old, you take drug for other ailments as well, right? And all these drugs interact with each other, because of which you have issues like neurological disorders, and also these drugs have side effects. And so we want to basically find a solution to this problem. And that's what I work on, right? Trying to find a cure for HIV.
Okay, so tell me about some of the original research you have conducted right throughout your career.
So during my PhD, as I said, that I was trying to make a vaccine for HIV, and the problem. So there are a few problems with HIV. One, why we don't have a vaccine for it yet. You think about COVID, right? COVID, within a year we had a vaccine and we started vaccinating everyone. So despite 40 years of research, why we do not have a vaccine for HIV yet? And the reason is HIV virus changes very quickly. It mutates really fast. Just like now we are seeing some of these COVID variants coming up, and the vaccine, sometimes what happens is the antibodies that are generated by vaccines, they are not good enough to prevent infection, although they still prevent serious disease. And that's for COVID. And the COVID virus mutates much slowly as compared to HIV virus. HIV mutates really, really fast, much faster than flu. And for flu, you need a vaccine every 6 months. So and HIV is very good at evading our immune system. So during my PhD, I was trying to understand how HIV evades our immune system and why our immune system is not able to control the virus on itself. For example, for COVID, so many people, they get infected, and even without vaccination, even without any drugs, they control the virus pretty well. But that's not true for HIV. So when during my PhD, I was studying this virus, I noticed that the virus has a coat of sugar on its surface. It's covered by sugar, and those sugars are derived from humans, basically from our genes. So they are not basically they are not a product of viral genes.
Okay.
So our immune system doesn't recognize those sugars, and the virus evades immune response with the help of those sugars. So we were trying to make some viral molecules without those sugars, so that when you use them as vaccine candidates, then you get a good antibody response against the virus. So what people in the field used to think at that time was that if you remove sugar from the virus cannot be infectious. It cannot take its three-dimensional shape, and that virus you will never be able to produce such a virus. It will not have that sugar coat.
Okay.
So during my PhD, by developing a computational method, I was able to design a HIV virus which didn't have most of the sugars on the surface. So it was exposed on multiple places, and that exposure makes such a virus more vulnerable. So when you use such a molecule for vaccine, you get better antibody response against the virus.
Okay. Does pineapple belong on pizza?
That's a very nice question. I think so, yes. I'm a vegetarian.
Okay.
And the more options I get on the pizza, the more happy I am.
Okay. What qualities do you believe make a great scientist?
This is a very good question. I often think about it. I think perseverance is the main quality that makes someone a good scientist, and curiosity. So science is often about trying same thing again and again with changing one or two variables.
Right.
So it can often get very frustrating, because if you do not get success in your first attempt, but then keep trying changing variables and trying to make something work, which no one has done before, it can get daunting. So I think perseverance, positivity, and just in general having curiosity about why something is not working, that's what helps.
Okay. What is one moment when you realized you knew you made the right career choice?
I feel that almost every day, actually, and the reason for that is I really enjoy solving problems. And when you're working on a difficult problem, like for example, trying to find a cure for HIV for AIDS, then there are challenges every day, and you solve those small challenges, and just those small victories, they make me realize that I chose the right career. It's intellectually very stimulating. It's very satisfying to know that what we're trying to do is something that can bring a change in the society. So I enjoy. I would not pick up any single moment. I would say that I just enjoy the journey. I just enjoy the process of science.
What was a tough time in your career, and how did it change you for the better?
So I would say that in science, you can have days, weeks, and months that can be really tough. And that usually happens when you are working on a project. Suppose you are working on a project, 12 hours every day for 6 months, and after 6 months, the experiment fails because of whatever reason, because the technology is not there yet, or because your hypothesis was wrong, or because some reagent was bad, or it can fail for many reasons. And I think the toughest thing I have faced was when I was doing an experiment with an HIV virus, and it was a long experiment that went on for almost 1.5 years. And after 1.5 years, I found out the hypothesis that I had was actually, it came out to be something else. Like I was thinking, okay, if I, for example, remove the sugars from the surface of virus, it will become less infectious. And I removed those sugars from virus, and the virus became more infectious. So like the hypothesis completely turned on its head. But then I started thinking, why did that happen? And actually that led to some really cool discoveries. So I would just say just regular failures in science. That's what makes it tough.
So you mentioned that you grew up in India. Describe a situation in which you utilize your multicultural skills to solve a problem.
So I grew up in Delhi, and my mother tongue is Hindi. And then I went to Bangalore for doing my master's and Ph.D. where people use Kannada. I'm married to a Maharashtrian who speaks Marathi, and I speak with people in English here. So I speak multiple languages. Now I can read and understand multiple languages. And I think this gives me an advantage, which I realized during COVID times, because when COVID started, people had so many doubts about the virus, whether it was man-made or it was natural, whether they should take the vaccine or not, whether it's a big conspiracy bias. So there were a lot of conspiracy theories going around, and I realized that people are really hesitant about getting vaccines. And I also realized that because I come from such a multicultural background, I have this leverage where I can go and reach out to these people and talk to them in a language in which they understand, and take my scientific knowledge there and clarify their doubts. So I think that's where I felt I made a real difference, because coming from a very different background, from a family where no one was really studying science, coming from that background, I understand their doubts. And then because of my science background, I have the ability to actually, answer some of their questions. So that's where I think I made a big difference, and then my multicultural background helped me.
What are your future goals in science?
Okay, so as you might guess, my biggest goal is to actually find a cure for HIV, and I seriously believe in it. If many of us together, put our mind into it, we might be able to find a cure for HIV. Professionally, I think once I finish my research in the Marson lab, I would like to move on to more independent position and study more viruses and get more into gene therapy.
Okay. What does your daily routine look like?
It's I don't know if I should say this in front of camera, but it's very hectic. It's basically, this is my daily routine look like. I come to the lab at around 9 a.m. And I'm here till almost dinner time, like 7 p.m. And then I go to the gym at 7 p.m. Then reach home at 8 or 8:30. And then my wife is also a scientist, so both of us, discuss some more science because both of us love it. And on the weekends, I go on hikes, I go for running, I go for biking. I love to explore San Francisco. That's it.
So now tell me about the strategy and the mindset behind your role. What does it take to be Ujjwal Rathore? So the way, so when you are a scientist, and
as far as your research is concerned, when the process involves many steps to solve a problem. Sometimes when people think about scientists, they think about someone must be just sitting in the lab and mixing many chemicals with each other. But I think as a scientist, a lot of time goes into thinking about the problem that you're trying to solve. That's where it starts, the scientific process. And then thinking about what are the unanswered questions, why those questions have not been answered yet. What can we do differently that we can find answers to these questions? But strategy-wise, a lot of thought also goes into who are other right people to bring into your team so that you can solve this difficult problem. So, for example, you must have heard earlier that Thomas Alva Edison discovered this, made this, invented this. But now we don't hear names of individual scientists that much. Because I think now the way we are making discovery is because of our collective intellectual wisdom and our collective efforts. So a lot of my time also goes into making collaborations, then also raising money for the research we do. So basically writing grants, writing fellowship applications, and then planning experiments, doing them, and doing data analysis and figuring out the results. The last step is usually to disseminate the results that we got by writing a scientific publication and telling what we have found to everyone.
What would you say is the most challenging part of your job?
As I said earlier, the most challenging part is just dealing with failures. So research is, I think, all about dealing with failures, because you try to solve a problem which has never been solved before, and try to work on something really which is cutting edge. So it's very common to get failures, and I think just dealing with that failures is the thing that you need to learn.
Do you think the sky really is the limit?
I don't know what that means, but if it means that you can grow as much as you can, I think that's possible in science. You can, anyone coming, I would say someone coming from public school in Delhi, studying Hindi medium, thinking about curing HIV. If who knows, I might. so I think sky's the limit. Yeah.
I believe in you, Ujjwal. What would you say is your greatest achievement as a research scientist so far?
This is a difficult question. I think what I feel really satisfied with is not just that I'm able to interact with people who actually discovered HIV in 1980s, but I'm also able to interact with people who are just coming out of high school, and I'm able to mentor them. So I think I feel a sense of accomplishment when I see that I can be that connection between someone who is really a well-accomplished scientist right now and someone who's just entering into the field of science and trying to understand it. I really enjoy mentoring, and so I feel really satisfied when people who I mentor, they go on to make big discoveries.
So, do you have any advice for the younger generation of scientists out there who may or may not be watching this video?
I think they are much smarter than me, but I would just tell them that science is all about finding joy in small things, in small discoveries. So don't be disheartened if you face failures in your experiments, or if you're curious, if you are watchful, you will always find something very interesting, which will not change the world around you. It will also change the way you look at things. And so I would say just be curious, be positive, and be collaborative. Work as a team. Discover things with your friends.
Thank you so much, Ujjwal. Ujjwal Rathore.
Nice to meet you.